Hello friends, hope you have enjoyed the previous talk containing overview of cytoskeleton. Here in this lecture we are going to learn microtubules as one of element of cytoskeleton. Microtubules are tubular structures involved in many aspects of cell structure, motility and transport. A microtubule consists of a hollow cylinder of tubulin subunits to which a variety of proteins may be attached. Notably, the microtubule based motor proteins and the microtubule associated proteins that play a more structural role. Here, we will mainly restrict ourselves to novel aspects of microtubule structure and mention other proteins only in this context. Although they are both proteins that help define cell structure and movement, they are very different molecules. So first, we will discuss the structure of microtubule and then we will focus on its functions. Let's talk about microtubules in more detail. Structure of microtubules. As we have studied previously in the overview of cytoskeleton, microtubules are the largest cytoskeletal components in cells with a diameter of 25 nanometers. Like microfilaments and intermediate filaments, microtubules are not randomly distributed in cells. Rather, microtubules radiate from the centrosome, which is the primary microtubule organizing center referred to as MTOC in animal cells. Microtubules are made up of subunits called tubulin. Each tubulin subunit is made up of one alpha and one beta tubulin that are attached to each other. So technically, tubulin is a heterodimer and not a monomer. In a microtubule structure, tubulin monomers are linked both at their ends and along their sides laterally. Microtubules are highly dynamic and will frequently elongate and shrink at a rapid yet constant rate. Here, formation of microtubules. Tubulin dimers can depolymerize as well as polymerize, and microtubules can undergo rapid cycles of assembly and disassembly. Microtubule polymerization occurs at plus end and depolymerization occurs at minus end. Dimers can be added very quickly, making it longer, or dimers could be taken off, making it shorter. So, it can become longer and shorter very quickly. During this phenomenon known as dynamic instability, tubulin subunits will associate from the plus end of the protofilament. A number of factors regulate the dynamics of microtubule formation. However, the primary determinant of whether microtubules elongates or shrink is the ability of GTP. GTP binds to the beta subunit of tubulin dimer that promotes polymerization and elongation of microtubules. Also, there are two different types of microtubule organizing centers. They are centrosomes and basal body. The centrosome is composed of centrioles plus other proteins. The other type of microtubule organizing center is called basal body. The basal bodies have either cilia or flagella. Cilia are these hair-like projections that come out of the cell. For example, cells of the respiratory tract have cilia and they have bit in an upward direction, and they helps to push mucus up our respiratory tract, and that's why you will sometimes cough up mucus. Flagellum is a tail-like projection that comes out of the cell, and it helps the cell move, and in fact, the only human cell that has a flagellum is the sperm cell. I would like to add here that flagellum of eukaryotic cells are different than that of the prokaryotic cells. So here we have a cell, we have a nucleus in the center, and right over here is blue is the microtubule organizing center or the basal body. Remember, It has same structure as centriole, and anchored to the basal body is a flagellum or a cilia. So, flagellum and cilia have pretty much the same structure. The only difference is that cilia tend to be shorter and flagella are longer. In cross section of cilia and flagella, both are composed of microtubules. Both of them are made up of pairs of microtubules, and they are actually 9 pairs. So, 1, 2, 3, 4, 5, 6, 7, 8, 9 pairs, and in the center there is one lone pair. So, that is why this arrangement is called the 9 plus 2 arrangement. Between the pairs of microtubules are protein called nexin. Nexin helps to keep the microtubules in their place and then coming out of the microtubules. We have a protein called dynein arm that breaks down ETPA and uses that energy to help the microtubules move past each other, and that is actually what drives the movement of the flagella and cilia. Formation of microtubules. Tubulin dimers can depolymerize as well as polymerize, and microtubules can undergo rapid cycles of assembly and disassembly. Microtubule polymerization occurs at plus end and depolymerization occurs at minus end. The rate of polymerization is generally more than the depolymerization, so the microtubule elongates. Beta tubulin bind GTP, which functions analogously to the ATP bound to actin to regulate polymerization. In particular, the GTP bound to the beta tubulin is hydrolyzed to GTP during or shortly after polymerization. This GTP hydrolysis weakens and binding affinity of tubulin for adjacent molecules, thereby favoring depolymerization and resulting in the dynamic behavior of microtubules. Like actin filaments, microtubules undergo treadmilling, a dynamic behavior in which tubulin molecules bound to GDP are continually lost from the minus end and replaced by the addition of tubulin molecules bound to GTP to the plus end of the same microtubule. In microtubules, GTP hydrolysis also results in the behavior known as dynamic instability, in which individual microtubules alternates between cycles of growth and shrinkage. Whether a microtubule grows or shrinks is determined by the rate of tubulin addition relatively to the rate of GTP hydrolysis. As long as new GTP bound tubulin molecules are added more rapidly than GTP is hydrolyzed, the microtubule retains a GTP cap at its plus end and microtubule elongation continues. However, if the rate of polymerization slows, the GTP bound to tubulin at the plus end of the microtubule will be hydrolyzed to GDP. If this occurs, the GDP bound tubulin will dissociate, resulting in rapid depolymerization and shrinkage of the microtubule. Functions of microtubules. Microtubules are the main building blocks that form the cytoskeleton, which is the cell's framework within which all components of the cell are held in position or allowed to move within certain constraints. Microtubules regulate cell division, cell morphology, intracellular transport, and axonal stability, and therefore play crucial roles in cell function. Let us study in detail function in cell division. Microtubules are known to have a great influence in the process of cell division. During cell division, dynamic microtubules radiating from spindle poles attached to chromosomes that the kinetochore and align them at the equator of cell. Spindle microtubules rapidly fluctuate between growing and shrinking until the chromosome is captured at its kinetochore. Taking a closer look, the presence of notable microtubules attached to the outer plate of kinetochore. Once captured, a combination of two mechanisms are thought to generate the assembly of chromosomes. First, polymerization and depolymerization of microtubules can push or pull the chromosome. In addition, positive and directed motor protein is thought to push the kinetochore away from the pole along the growing microtubule, and negative and directed motor proteins such as dynein could pull the kinetochore towards the pole and greater kinetochore to shrink microtubule. Microtubules from the opposite pole captures the sister kinetochore and chromosomes are eventually aligned at the equator. Function in cell morphology. Maintenance of elongated shape and polarized pseudopodial activity of many cultured cell types including fibroblasts and neurons depends on the presence of an intact microtubule system. Alternatively, microtubules running parallel to stable cell edges may act as a rigid structural element which supports cell asymmetry by directly counteracting compression mediated by the actin cytoskeleton. Function in transport within the intracellular transport. Involvement of microtubules in intracellular transport of organelles has been a subject of recent extensive studies, and microtubule dependent motor proteins are thought to be responsible for organelle transport along microtubules. One of these proteins, kinesin and dynein, appears to be the most abundant translocator responsible for microtubule dependent transport toward the cell periphery. Kinesin and dynein help to shuttle different things from the soma down to the axon or the synaptic terminal. For example, synaptic vesicles which actually contain neurotransmitters move to the axon terminals. Different lipids that might be necessary and even organelles such as mitochondria as well as kinesin and dynein are able to transport substances in this direction from the soma to the synaptic terminal. This process is called axonal transport or exoplasmic transport. Microtubule dynamics in neurons is modulated by several accessory proteins termed microtubule associated proteins. An important microtubule associated protein is the protein tau, which promotes formation of axonal microtubules, stabilizes them and drives neurite outgrowth. Impaired interaction of tau with microtubules plays an important role in the pathology of several neurodegenerative diseases. Dysregulation by genetic mutation or hyperphosphorylation affects the tau microtubule complex, leads to tau detachment, causes instability and disassembly of microtubules and thus perturbs axonal transport. Dysregulation by genetic mutation or hyperphosphorylation affects the tau microtubule complex, leads to tau detachment, causes instability and disassembly of microtubules and thus perturbs axonal transport. Microtubule stabilizing drugs might therefore improve neuronal degeneration. Drugs that affect functioning of microtubules. A wide variety of drugs are able to bind to tubulin and modify its assembly properties. These drugs can have an effect at intracellular concentrations much lower than that of tubulin. This interference with microtubule dynamics can have the effect of stopping a cell's cell cycle and can lead to programmed cell death or apoptosis. However, there are data to suggest that interference of microtubule dynamics is insufficient to block the cells undergoing mitosis. These studies have demonstrated that suppressions of dynamics occurs by concentrations lower than those needed to block mitosis. These studies have demonstrated that suppression of dynamics occurs at concentrations lower than those needed to block mitosis. Suppression of microtubule dynamics by tubulin mutations or by drug treatment have been shown to inhibit cell migration. Both microtubule stabilizers and destabilizers can suppress microtubule dynamics. The drugs that can alter microtubule dynamics include the cancer-fighting taxane class of drugs called paclitaxel, also called Taxol, and docetaxel from the yew tree block dynamic instability by stabilizing GDP-bound tubulin in the microtubule. Thus, even when hydrolysis of GTP reaches the tip of the microtubule, there is no depolymerization and the microtubule does not shrink back. And thus, these drugs are used in the treatment of breast, lung, and ovarian cancer. The apothilons, for example, ixabepilone, work in a similar way to the taxones. Nocardazone, vincristine, vinblastine from Catharanthus roseus, and colchicine have the opposite effect, blocking the polymerization of tubulin into microtubules. Eribulin binds to the positive growing end of the microtubules. Eribulin exerts its anti-cancer effects by triggering apoptosis of the cancer cells following prolonged and irreversible mitotic blockade. Expression of beta-tubulin of the type 3 has been reported to alter cellular responses to drug-induced suppression by microtubule dynamics. In general, the dynamics are normally suppressed by low subtoxic concentrations of microtubule drugs that also inhibit cell migration. However, incorporating beta-tubulin into microtubules increases the concentration of drug that is needed to suppress dynamics and inhibit cell migration. Thus, tumors that express beta-tubulin are not only resistant to the cytotoxic effects of microtubule targeted drugs, but also to their ability to suppress tumor metastasis. Role of calcium ion. Microtubule polymers are extremely sensitive to various environmental effects. Very low level of free calcium can destabilize microtubules, and this prevented early researchers from studying the polymer in vitro. Cold temperatures also cause rapid depolymerization of microtubules. In contrast, heavy water promotes microtubule polymer stability. Pathological conditions. Let us study the abnormal condition that occurs in association with dysfunction in microtubules. As discussed, microtubules are a component of cytoskeleton and play a central role in organelle transport, axonal elongation, and cellular architecture, especially in neurons. The polymerization, stabilization, and arrangement of microtubules can be modulated by interaction with a series of microtubule associated proteins referred to as MAPs. Recent studies have proposed the abnormal alterations of two major microtubule associated proteins, tau and MAP2, in the brain tissue of naturally occurred and experimental human and animal prion diseases. Increased total tau protein and hyperphosphorylation of tau at multiple residues are observed at the terminal stage of prion disease. The abnormal aggregation of tau protein disturbs its binding ability to microtubules and affects the microtubule dynamic. This protein family leads to disruption of microtubule structure and impairment of axonal These protein family leads to disruption of microtubule structure and impairment of axonal transport and eventually triggers apoptosis in neurons, which becomes an essential pathway for prion to induce the neuropathology. Summary. Coming to the summary of today's lecture, we have gone through the structural integrity aspect of microtubules as the structure so obtained from tubulin heteromers that polymerizes into protofilaments and associate laterally as an assembled structure. We included in our discussion part the functions associated cell morphology, intracellular transport and axonal stability along with the major role in mitosis process or cell division. We have lastly also learned an important microtubule associated protein called the MAP2 microtubule associate protein and tau protein, and also studied its role that any dysfunction in them would cause a serious pathological conditions. Thanks.